So yeah, this is Park. I'm working for 대구경북 Medical Innovation Foundation, which is called K-medihub recently, and Medical Device Development Center. And my title today is the principle and application of CRISPR diagnosis. And my recent research data will be shown, which can be, that data is related to this CRISPR diagnosis. So my research data will be open here. This is contents. And firstly, I'm gonna talk about the principle of CRISPR and CRISPR-Cas diagnosis. And I will review some application of CRISPR-Cas diagnosis. And my recent data about Aptamer CRISPR-Cas and LAMP CRISPR-Cas for VHSV, my target, will be discussed. And these two topics, these ongoing topics, are not finished yet. So please understand that some part of data is a little bit hidden in the slide. Yeah, this is principle of CRISPR-Cas. In the CRISPR-Cas system, the most important part is this. Can you see this cursor? Is it moving in this slide? Yeah. This ribonucleoprotein is the most important part, which is consists of this RNA part and this Cas protein part. So when this ribonucleoprotein identifies a target gene, this target gene can be called as protospacer, and this RNA is called spacer. Actually, this RNA is divided into two parts. One is a spacer, and the other one is a tracker RNA. So this spacer is related to the recognition of this target gene, and this tracker RNA is responsible for the binding to this Cas protein. So when this ribonucleoprotein complex recognizes this target gene, this whole ribonucleoprotein is activated, and this activated ribonucleoprotein is cleaved its target. So this guide RNA can be designed to be complementary to this target gene. Then this Cas system can be used for many purposes. And one more thing you need to consider is that this PAM stands for protospacer adjacent motif. So I mean, this PAM should be on the target gene. Otherwise, this Cas protein cannot recognize this target. And this PAM sequence is different from this Cas protein. So at the beginning point, the PAM should be considered. That is the CRISPR. And the next part is CRISPR-Cas diagnosis. This CRISPR-Cas diagnosis begins with the discovery of additional function of this Cas protein. As it is known, this enzymatic cleavage occurs when this conjugate of Cas protein meet or detect is a specific complementary sequence. But its important part is that enzymatic cleavage occurs not only on this target sequence, but all this surrounding non-specific single-stranded sequence is also cleavaged. So for example, in this probe is modified with fluorescent molecule and potential. So this probe cannot make a fluorescent signal. But when it is cleaved like this, then this fluorescent signals turned on again. So this principle is used for many CRISPR-Cas diagnosis. And one of the most important the famous part is this Sherlock Bioscience. I don't know if another person heard about this company, but it is the most famous company in this CRISPR-Cas diagnosis, and they have FDA approval this detection kit, Sherlock CRISPR SARS-CoV-2 kit. And the second famous company is Mammos Bioscience. I heard that Professor Daudena is related to this. Professor Daudena is, she is 2020 Nobel winner in this part. Yeah. So this CRISPR-Cas diagnosis is used for many various detection systems. So from this slide, I'm going to talk about some application of CRISPR-Cas diagnosis. So firstly, this is gold nanoparticle-based colorimetric CRISPR sensor. In this concept, this linker DNA and the target is incubated with Cas system. And this linker DNA is designed to make aggregation of gold nanoparticle like this and this. So when there is target, this Cas system clears all these single-stranded linker, not only target, but also all this linker. So there's not any aggregation on gold nanoparticles. That means there's no aggregation when there's target chain. So by measuring the absorbance at around 520 nanometer, it is possible to determine the correlation between the concentration of target and its color change. Of course, on the other hand, when there's not any target, it makes this aggregation. So its color should be blue like this. A very small concept. And the next one is electrochemical CRISPR sensor. So the authors named it Ecrispr sensor. So in this Ecrispr sensor, this probe, single-stranded probe, is used, and this single-stranded probe is modified with methylene blue and is immobilized on this gold surface. So after loading the target and the CRISPR, consequently, the signal change is analyzed. If the target DNA is present, the Cas complex will be activated to cut the probe. So this methylene blue can be released. That is sort of enhancement of this signal. So when there is a target, this signal is there, but in case of CRISPR-Cas, this methylene blue is released, so it makes some signal enhancement like this. So its limit of detection is known to be 50 picomolar in this system without any amplification. I think it's quite high, quite good, I mean. And next one is a very interesting microfluidic chamber, which use this CRISPR-Cas system and this magnetic bead and platinum nanoparticles. Firstly, this platinum nanoparticle is linked to this big magnetic particle with this single DNA. When there's a target in this chamber, this Cas system cleared the single strand of DNA, and this platinum nanoparticle is released. Then this released platinum particle go to this second chamber, and is catalyze the generation of oxygen from this hydrogen peroxide. This oxygen increases the pressure of this microfluidic chamber, and it pushes this detection ink. So in case there is some target, it makes some oxygen, and this oxygen pushes the ink. So this moving of ink is sort of the detection signal. So with this detection signal, they said that it is possible to detect 10 picomolar of the target. With this microfluidic chamber, some cell-free DNA in serum and cancer-related mutant DNA was detected. Yeah, like this, they have developed a lot of interesting CAS-based diagnostic system. So from this slide, I'm going to talk about my research, which focused on the screening of aptamer. This is the concept of the aptamer CRISPR system. The main advantage of this aptamer CRISPR system is that it is possible to detect protein biomarker instead of nucleic acid. So here, the aptamer sequence should be linked to the target sequence that is recognized by this ribonucleic protein. And for the designing of this aptamer and the target sequence, the important part of this link is that the aptamer structure should be maintained after it is linked to the target sequence. So firstly, the microbead with this primary aptamer can capture this target protein. And after this capturing, this microbead and target is injected to this acoustophoresis system. In this acoustophoresis, micro-sized bead or any particle with micro-size is affected by acoustic wave, and it is moved to the center of the channel. The focused bead that can be collected, and this CRISPR-Cas system recognize the second aptamer and target sequence. So this recognition can make this additional cleavage, and this principle can make this fluorescence signal. That is my plan. And that's the movie I made for the separation of larger microbead from this small microbead. And this is about the Scientex method. I think in order to make the aptamer CRISPR system, the important part is that it should be designed the short sequence aptamer to avoid the other secondary structure with this target sequence. So to develop the short aptamer, this primer lesion was shielded. So the shielded library was incubated with this target. Then this exposed part is working to make this structure. But this primer lesion, which is shielded, can be used for amplification. And after finishing this process, this whole sequence was sequenced, and this random part can be only synthesized and used. This is the strategy to make short sequence. And yeah, after 10 times repeating, 11 candidates were selected, and these 11 candidates have 40 base pair, and this 40 base pair sequences were analyzed with SPR, and this sequence was analyzed for the specificity and the affinity. So for the analysis of specificity, the main target and 3 other counter targets are analyzed, and for the affinity, 5 different concentration of target was analyzed with SPR. Yeah, this is the data of the specificity of candidates. As it is shown, out of 11 sequences, this 7.1 and 7.2 and 10.1 shows the higher specificity. And this is the plotted data of the affinity. And as it is shown, 3 candidates are showing quite reasonable affinity, and these 3 candidates are linked to the target sequence. I think this slide is the most important slide for this abnormal study. This is the whole sequence of this 7-1, and this part is the linked to the target sequence for the CRISPR-Cas recognition. As it is shown, this structure was not affected with this target sequence. And in case of the 7-2, it is also this main structure of 7-2 is not affected with this target sequence. So I think that this 7-1 and 7-2 can be used for CRISPR-Cas system. But in case of 10-1, this structure is not there anymore. So it is affected by target sequence. So this S10-1 is abandoned, I think. So further, this 7-1 and 7-2 will be studied for CRISPR-Cas diagnosis system. The next part is about the isothermal application with the lamp. Yeah, as it is shown in this table, some Cas, this is the name of CRISPR diagnosis. This method uses a pre-amplification process to maximize the sensitivity. So I also plan to make some pre-amplification method before applying this CRISPR diagnosis. Actually, there are many methods for isothermal amplification strategy, but this LAMP and RPA is known to be better than another method in terms of reproducibility and its specificity. And compared to RPA, this LAMP is actually more complicated to design the primers, but its specificity is known to be better than this RPA. So I decided to use this LAMP strategy. And in this study, the target is viral hemorrhagic septicemia virus, which is highly contagious virus leading to high mortality to these fishes. So to test this LAMP primer, RNA virus was extracted from this splint part. And yeah. From now, some data is hidden because it is under patent applying now. But I think you can see this band or something. So first of all, after design of the primer for LAMP, the optimizing temperature is tested. As a result, it is turned out that at least 62 degrees are required to amplification. The result from this electrophoresis is well matched to the result from this fluorescent result. So this temperature and this time is decided to be the condition of this LAMP. And the next part is limit of detection. So with the condition of 65 degrees and 30 minutes, the various diluent was tested for limit of detection. So with this condition, it is turned out the 55 femtomolar. Yeah, I think it is reasonable to detect this virus. And the other part is, the next part is colorimetry part, but it took quite a long time to detect this femtomolar. I think it is because it took longer longer time for a change of pH when it is increased during this lamp procedure. So I don't think this colorimetric method can be used for this lamp. But yeah, if the time is not important, but it can be also used. The next is the final slide of my data. And it is real-time lamp because it is also fluorescent-based signal. It is as sensitive as normal fluorescent method. Its limit of detection is around 30 femtomolar. So I think this method is also possible to use to detect the VHSV with this primer. So this primer set will be studied for the CRISPR-Cas diagnosis, but the next part is not done yet. So it will be soon performed. And this is the end of my ongoing project. And from now on, I would like to introduce my institute very briefly for 1 or 2 minutes. This Daegu Medical Innovation Foundation is located in this red dot in Daegu. And Daegu is well known to be a medical city. And we have 4 centers like this Clinical Drug Manufacturing Center and New Drug Development Center and Preclinical Research Center and Medical Device Center. I'm working for this Medical Device Center to develop many diagnosis systems. And yeah, all these 4 centers are working together to, I think, to support this medical market or many research, a lot of things related to the medical business. And this medical device center, of course, focusing on this research and development, but another people are doing for the producing the prototype with very high resolution 3D printer and PCB, and this evaluation of the equipment and evaluation of this sample of toxicity, and the technical document can be evaluated in this center. So there are a lot of equipment like MRI, so PET, and the other many equipment for the analysis. So many companies are trying to use this equipment, and we have a lot of certification. So I hope you also can use this facility and this evaluating system.